Charles Darwin says that diversity is key to success. Welcome to this webinar about mining precision nutrition. And indeed, we want to mine the gut microbiota, an environment where diversity is key as well. My name is Isabel Gunnings. I'm the Scientific Program Director at ILSI Europe. And welcome to this webinar. ILSI Europe, for those who don't know it yet, is a non-profit organization. And our mission is to advance and generate emerging science and research to ensure that food is safe, nutritious, and to pay attention to the planetary and human health and well-being. Key for this mission is our tripartite model. We want to create collaboration between experts from industry, academia, and the public sector. And this webinar is supported by the Pre- and the Probiotics Task Force. And as you can see in this task forces, we bring together experts from the public and private sector. The classical deliverables of ILSI Europe are peer-reviewed publications, as you can see here on the screen. Since the start in 2010, those two task forces generated over 13 papers, and one of the latest one you can see here on the screen. If you want to see the full list, and all publications are open access, you can join our website. Besides that, the task force also generates guiding documents and educational materials, and I definitely invite you to have a look at our recent monograph on the dietary probiotics and the gut microbiota in human health. It has been translated in about 7 languages, so I think we can find one version that suits you best. And then, of course, ILSI doesn't stop at just generating science, it also communicates and disseminates it, just like this webinar today, where we are talking about precision nutrition. About this webinar, recordings will be made available to you on the website. We would like and appreciate a lot if you can give us some feedback about the content and also the organization, so you will receive a questionnaire after the webinar itself. If this webinar tastes for more and you want to know more about what the activities of the Pre- and Probiotics Task Force, I already invite you to another webinar on the 29th of November, where we will do a call for interest and we'll share with everybody new activities that are coming up. So definitely mark your agenda on this. And with no further ado, that some of the seeds are planted in your brain, that you maybe find a tool that fits you and your research best. So with this, I hand it over to our chair of today.
Thank you, Isabel. Thank you for the introduction. Good afternoon, good morning, wherever you are, and thank you all for joining today. My name is Gabriele Gross. I'm very happy to be chairing today's webinar. I'm a part of the Science Platforms Group within R&D at Reckitt Johnson Nutrition, and I'm one of the industry member representatives in the ILSI Europe Probiotics Task Force. I have had the pleasure to work with a recent expert group linked to ILSI Europe, and I would just like to very briefly share the context of that activity with you as a background to today's webinar before we head right into the presentations. So with that expert group, we have started actually with a challenge that in many cases it can be difficult to reproduce clinical trials, not only with probiotics, but also other nutritional interventions due to the variation in responses between individuals. And we were wondering in how far these differences in gut microbiota contribute actually to these individual responses. We were wondering what are current tools available to study and measure these, and how could such approaches be leveraged to optimize precision nutrition and healthcare in the future. And this is actually what we want to talk about today. So the insights from this expert group has been described in a recent scientific publication that will be presented to you today by Dr. Sean Gibbons. He is the third speaker in our webinar. And before that, we will have 2 other presentations, one on the importance of the gut microbiome for personalized nutrition by Dr. Emily Leaning. And we will kick off with the first speaker today, Dr. Gwen Foloni, to set the scene on gut microbiome research. Just for housekeeping, we have structured the webinar such that these presentations will be approximately 20 minutes each, and there will be short time in between the presentations to address some immediate questions that you might have. But we will have time in the end for overall questions and also a brief discussion with the experts at the end of the webinar. So please do add your questions in the chat function so that I can take them along in the Q&A and the discussion sections. And we are very curious to hear your comments and thoughts on the topic. So please use the chat function. So now to start, it's my pleasure to introduce our first speaker today, Dr. Graham Faloni. He's a staff scientist at the Flemish Institute for Biotechnology in Belgium. As one of the architects of the Flemish Gut Flora project, his work focuses on defining the boundaries of a health-associated gut microbiota. And today he will talk about quantitative microbiome profiling and community type analysis in health and disease. The floor is yours, Gwen.
Thank you very much, Gabrielle. Can you just confirm that you're hearing me well?
Yes.
Okay. So thank you and good afternoon, everybody. I'll start my presentation. I'll start my presentation just with introducing you to the human microbiome. You might have heard the statement that human beings are not just human beings, but that we actually carry 10 times more bacteria than we carry human cells. That's an interesting observation. It's not always true. It depends a bit on the time of the day that you would be measuring or you would be calculating the numbers of cells that you're carrying around. And why is that? Well, that's because most of the bacterial cells that you have or that you possess, they're actually located inside your large intestine, and that one or more times a day you get rid of many of those. That changes the balance a lot. It will go from 1 to 10 to 1 on 1 actually. So most of the human microbiota or the human associated microbiota is located in the gut, and it has been studied for several decades actually. And what do we know about this microbiota? Well, we know that it expands our digestive capacity, actually degrades the things that we cannot digest. It produces some essential nutrients and vitamins, like, for example, vitamin K. It increases the colonization resistance against pathogenic intruders, which just means like if there are good guys living in your gut, there is no room for the bad guys. And it assists in the detoxification of some xenobiotics. Now, for a very long time, we've been studying this gut microbiota, this community, by cultivation-dependent techniques, like you see here. This is already microbial genomics, but in the early days, we needed to isolate individual bacteria, cultivate them, grow them, get out the DNA, and then reassemble the genome of the bacteria that we were studying. Nowadays, we don't do that anymore. We use a technique called metagenomics. We just take an environmental sample, which here would be a poo sample. We prepare the DNA, we sequence randomly, and that gives you a giant puzzle. But with the necessary computational power, you can actually reassemble that puzzle and get parts of the genomes of the bacteria living in the colon or living or present in the feces. And by analyzing these partially assembled genomes, you can actually deduce which bacteria that are living in the feces or in the gut and what they would be capable of. And why are we analyzing this microbiota? Well, what's your poo telling you? We want to move forward, or that's one of the aims at least, move forward to health monitoring, screening of health through microbiota analysis. And one step further would, of course, be to modulate an individual's health by changing his gut microbiota. But unfortunately, we're not there yet. So my contribution of our lab to this endeavor has been the Flemish Gut Flora project, where we have been trying to establish the boundaries of a healthy gut microbiota in Flanders, in Belgium. It's a small region with around 6 million inhabitants. And we've done that under the umbrella of the Flemish Gut Flora project. Now, the Flemish Gut Flora project are actually several projects. There is the original one, which started in 2014, where we sampled the microbiota of around 3,000 individuals. Later on, we moved forwards with the 150 Days of Gut Flora project, where we are following 350 individuals longitudinally during 150 days. 1 month they had to provide a daily sample, actually, so it was a quite intensive project. And more recently, we completed the 5-year later project in 2019, where we reassessed the microbiota of the participants of the original project in 2013. But today I will mostly speak about the 2014 project, because for that project we have completed, or mostly completed, nearly completed all the analysis, and that's also the project that we have been publishing about in recent years. So let's take a look at the results on the microbiota variation in Flanders. So this graph is called the PCOA. Every dot here actually represents a microbiome of an individual, and the distance between the dots corresponds with the distance between the microbiome of that individual. So what can we learn about this microbial diversity here? Well, the first thing that we looked at, and that's mostly because of historical reasons, is at enterotyping. And when you're doing enterotyping, you're looking at community types, at specific microbiome communities that are shared by several individuals. And well, there has been some scientific debate about the concept of enterotyping, but I mean, the methodology has advanced a lot during the last years. And with this presentation, I hope to convince you that it's really a valuable methodology to follow. So here in the Flemish Gut Flora project, we managed to identify 4 different community types, which we named after the dominant species. So the green one is the Prevotella community type, the yellow one, the Rhymnococcaceae community type, and then there is the blue one, Bacteroides 1, and the red one is the Bacteroides 2. Bacteroides 2 is a more particular, specific constellation. I will talk about that a little bit more in a few minutes. Next, what we did is we took a look at those factors that are actually driving microbiome variation, at least in Flanders or in people living on a Western diet. And to do that, we constructed a set of metadata variables. In fact, we had 503 metadata variables that we then associated with microbiome variation. And we found that 96 among them could correlate with microbiome composition. And 11 of them had actually a non-redundant contribution. So these ones are the ones that we call the microbiome covariates. But together, they were only explaining something like 4 or 5% of microbiome variation, meaning that 96%, 95% remained unexplained. Why is that? Well, there are several reasons. A big part of that is just inter-individual variation. But also this is just a cross-sectional study. I'm sure that if we would be doing a longitudinal study or looking at the results of a longitudinal analysis, we would be able to identify a lot more. And why is that? Because actually when you're looking at cross-sectional data, the food data, they become an individual fingerprint, because everybody's diet is quite unique. It's hard to find trends on diet in cross-sectional data. So they inflate the dietary data, inflate the inter-individual variation. But let's have a look at the results. We find some of the usual suspects here, the usual microbiome covariates like age, like gender. Here is also BMI. For the food data, we found the link with fruit consumption, but also with bread type preference. Of course, there's the impact of medication. These are the blue ones, like, for example, antibiotic intake. But the factor that contributed most to microbiome variation is actually the Bristol stool score. I don't know if all of you are familiar with the Bristol stool score, but the Bristol stool score is a score that allows you or allows a participant to assess the variation in stool consistency by these drawings here. So there are 7 scales that goes from very hard lumps to just a liquid stool. And Bristol stool score is very interesting because it links to stool consistency, and stool consistency links to transit time. And transit time is a key variable of the gut microbiota variation. It's influenced by the host, by genetics. It's influenced by lifestyle and drugs. It has an effect on the microbiota, but the microbiota can also have an effect on transit time. So the transit time actually imposes a dilution rate on the colon, meaning it determines which bacteria will get washed out and which are allowed or can stay. It will also determine nutrient limitations. If you have a long transit time, nutrients in your bowel will be depleted, so the bacteria will have less food, and they will have to change their metabolism. But it also drives end product accumulation, and those end products, or some of these end products, can definitely influence the transit time also. So we call it a bit forgotten covariate. Here, this picture might give you the idea that it has a lot to do with dilution, and that's also a bit the case, but not completely, yeah, because you see, the stool consistency also links to community type variation. For example, in the hard stools, we find a lot of the Rhymnococcaceae enterotype, while the Prevotella enterotype is more present in the loose stools. Stool consistency and transit time also linked to richness. In hard stools, very hard stools, you find a higher richness, and in liquid stools, richness is a lot lower. So next, we added a quantitative layer to microbiome variation. What you see here is our profiles that you would normally get from a sequencing effort. So when you're sequencing stool samples, and you will see that all these data are actually proportional data. They are relative data. So you get actually the fractions of bacteria that are together making up the stool samples. Now, we wanted to move to another level, a quantitative level. So we decided to count actually the number of bacteria that we found in stool samples. We just did it with flow cytometry. And what you can see here is that there is definitely a correlation with the stool consistency or the stool moisture. Right now, if you have a high stool moisture, you have a low number of bacteria. But again, it's not only that. We see that the bacterial counts are different between the enterotypes. And there is one enterotype, and I told you before that it was a specific one. It's the Bacteroides 2 enterotype that seems to have a very low cell count. So it's a low cell count enterotype. And here, this is are some observations from a patient group. But even if we are correcting for stool moisture, so it would be because I told you that also the Prevotella enterotype is characterized or is more prevalent in loose stools. But even when correcting for stool moisture, you still see that this Bacteroides 2 enterotype has a lot less cells than you would expect. So it's really different than the other constellations. So once you have these counts, you can actually transform this matrix, the proportional matrix, to a quantitative matrix. And when you do that, well, of course, it has a huge impact on all the associations that you would do between bacteria or bacterial genera and covariates, or with, for example, bacterial genera and how they change upon a dietary intervention. I'm not saying that relative or proportional data don't have their value. They have their value, definitely. And if you're looking for biomarkers for disease or something, I would actually suggest that you stay with relative profiles. But if you're looking at effects and causes and consequences of interventions, then I would definitely go to the quantitative data. Based on all these results, we came up with this model or this ecological framework to look at microbiome variation in healthy individuals, although it can also be used for patient groups, depending on which disease that you're looking at. So here you see the link between the stool consistency or the transit time and what you can expect to find in your stool sample in the microbiota. So actually, we consider the stool as an ecosystem that has been maturing in the bowel, and the composition and the metabolic activity actually depends on the moment of ingestion. And so how long the stool has been maturing, or the food matrix has been maturing in your colon. If you have loose stools, so that means that you have a fast transit, you'll have a high water content in the stool, a low microbial density. And because of the short transit times, the bacteria will mostly focus on saccharolytic activities, right? And here at the bottom, it's the richness. You still have a low richness in those stool samples. And at the other end of the spectrum, you'll find the hard samples. Hard samples have a low water content, a high microbial density. The bacterial metabolism will shift from saccharolytic to proteolytic metabolism because all the readily fermentable substrates are gone already, and the richness will be a lot higher. So here we look at richness actually as a marker of an ecosystem development. It has previously been proposed to be a marker of health, and that's, generally speaking, that's true. So that's an association that's correct, but I think it's even more correct to see it as a marker of ecosystem development. But these two things, they go hand in hand. Now let's have another look at this Bacteroides 2 enterotype. This Bacteroides 2 enterotype is the weird one, the one with the low cell counts. After observing that it was aberrant, we started to looking at the prevalence of this enterotype in different patient groups. And here is an example of this analysis. You see that in healthy populations, you have between 15 and in some cases even 5% of individuals that carry the Bacteroides 2 enterotype. But if you look to patients group, like for example here, ulcerative colitis patients, they have around 40%. But Crohn's disease patients, they carry around 80% of this aberrant enterotype. Now, this made us to, or we hypothesized then that this enterotype can actually be considered as a dysbiotic enterotype. So we had to define dysbiosis, of course, because there are several definitions of dysbiosis. So what we did is propose these definitions. We proposed it to be a microbiota community configuration that fails to perform its commensal role and contributes to disease onset or progression by increasing host susceptibility towards disease development, or by co-initiating or sustaining physiological processes affecting host health. Now, this is a slightly different definition than mostly handled, because mostly dysbiosis is a self-defining concept. So the community configurations found in patients group are called dysbiotic, but here this is not the case. We actually acknowledge that you could find dysbiosis in healthy individuals, but that it might have an effect on host health on the longer term, because it might make you more susceptible for disease development. This is a working hypothesis. We're still examining this. We are making progress on that, but we haven't published these findings yet, the related findings yet. So if this Bacteroides 2 enterotype would be a dysbiotic one, how would you get it? And maybe more importantly, how would you get rid of it? Well, what we believe, and also this is a working hypothesis, is that development of dysbiosis can be traced back to the what we call the main symbiosis in the gut ecosystem. It's actually the one between the butyrate producers and the colonocytes. So in an anaerobic colon environment, butyrate producers that are really sensitive to oxygen, they produce butyrate, and that's used by your colonocyte as their preferential energy source. And by doing so, they consume O2, oxygen, and they keep the gut environment anaerobic. Now, this equilibrium can be broken both at the microbiota site or at the host site, but the effect is always the same. The environment or the gut lumen becomes less anaerobic. The butyrate producers, they don't have a selective advantage anymore, and they stop producing butyrate, or even they die, or they become less abundant, and the metabolism of the colonocytes changes. And that's the moment that you get into a more dysbiotic ecosystem. And how can you break that equilibrium? I think there are several ways of doing so. Might be due to lifestyle and dietary habits, infection, inflammation, antibiotic treatment, and so on. Here, for example, you see the effect of a dietary intervention. It's a dietary intervention that we did in 20 individuals. This is only one individual. And it's a 3-week intervention. So in the first week, you have people were consuming their normal diet. Then came the intervention week, and then the 3rd week again, they switched to their normal diet. The intervention week, I cannot give you too much details about it, but it was a restricted diet with a limited number of food ingredients that they could consume. So from the moment that the intervention starts, you see that the cell counts, because these are quantitative data, are lowering. So the amount of cells we find in the stools are lowering. And it's not in general that they're lowering. The filum or the genus that reduces, what is reduced in abundance is the Fecalibacterium genus. And the Fecalibacterium is the one here in blue at the bottom. And you can see that it nearly disappears completely at the moment that we start with intervention. And Fecalibacterium is one of the most important butyrate producers in the human colon. So at the end of the intervention, you also see that the enterotype switches. So here on the top, you see the enterotype colors. This person had a Rhinococcaceae enterotype until day 4 of the intervention. And then, due to the intervention, he switched to a Bacteroides 2 in enterotype, the one that we call dysbiotic. Luckily, after the intervention stopped, and only 1 day after it stopped, he returned to his or her original configuration, being the Bacteroides 2, the Rhinococcaceae enterotype. So here you can see that with dietary interventions, you can definitely and strongly modulate the gut microbial system or ecosystem, in this case, towards dysbiosis. Here's another example of modulation of the ecosystem, and this is an observation that we made in a cohort of obese individuals, so not only of obese individuals, it had a gradient of obesity. So in lean individuals, we noticed that the percentage of Bacteroides 2 that we were observing was around 4%. This is lower than in the other data set, but mostly because the sequencing method here is different. This is metagenomic sequencing. But in obese individuals, we noticed that this went up to 17–18%. So there was an increase from 4% to 18% along a gradient of obesity. And what we noted then is that this increase was actually absent in a group of individuals that were taking statins. So statins, in this data set at least, because other people have published other data sets where they could not reproduce this data, we saw that statins had a modulatory effect or a potential modulatory effect on the gut microbiota composition by actually maintaining a eubiotic system. We could validate these findings in 2 independent data sets, but as I said, for the moment, there is some discussion about this. but we're actually performing some intervention trials to investigate this effect with more detail. So is there another way of modulating the gut microbiota to therapeutic interventions with statins? So, I seem to be blocked. Yep, no. The take-home message of my presentation of today. I think you need to consider fecal samples as a snapshot of an ecosystem under development. And in development in healthy individuals, it's mostly dependent to, or it's mostly linked to transit time. So I think that's a really important take-home if you're setting up studies or dietary interventions. You need to know what the transit time is of these individuals, and you need to assess whether the effect that you're seeing is not completely due to a change in transit time. Definitely with fibers, this might be an important factor. So not all healthy individuals have a eubiotic microbiota. But what we have seen, if we are right with our hypothesis that the Bacteroides 2 enterotype is dysbiotic, then we see that there is at least 5 to 15% of the healthy individuals that are carrying a dysbiotic microbiota, and that might have consequences on the longer term, but we are not sure of that yet. What we are sure of, or more sure of, is that not all patients have dysbiotic microbiota. We think that a dysbiotic microbiota might be a symptom of a lot of diseases, that prevalence of dysbiosis might change between diseases, but that you cannot just say that all patients have a dysbiotic microbiota. And we have also seen that dietary interventions can trigger dysbiosis, but most probably they can also be designed to regain and maintain a eubiotic ecosystem. And that brings me to the end of my presentation. So I hope you enjoyed it, and I will try to answer the questions that you might have.
Thank you very much for this presentation and amazing overview and sharing a lot of learnings with us. There has only been one question added to the chat, and I think probably that's what you already answered in the presentation. Did the samples come from feces, shed bacteria, or are they invasive sampling? Have you also been looking into other?
No, for the moment we only work with feces. Our protocols, because we work with so large patients group, we need to do home sampling protocols. So that is nearly impossible then to get samples from inside the large intestine. So yeah, this is all fecal material.
Yeah, that's very clear. Thank you. And there's just another one coming in. That's very timely. Excellent. Several publications do not show a change on enterotypes after diet. How do you explain the rapid changes in your population?
It is the diet itself. So it's the nature of the diet. This is a restriction diet where people were eating, they had to choose between 3 food ingredients and could only eat that during an entire week. So that's a very drastic intervention.
Okay, that's good to keep in mind what type of intervention indeed we are looking at in these specific examples. Yeah. Excellent. Thank you so much. I think we'll leave it with the questions for now and come back also in the joint discussion at the end of the webinar. Thank you so much for your presentation. And we will continue with our second speaker, Dr. Emily Leeming. She is a registered dietitian and nutrition scientist at King's College London, and Zoe, a personalized nutrition company. Zoe runs the PREDICT studies investigating personalized impact of food on metabolic parameters and gut microbiome. And Emily's presentation today is titled Microbiome, a piece of the personalized nutrition puzzle. Emily, the floor is yours.
Hi, everybody. Really great to be here, and really interesting talk by Gwen. So that was always interesting to hear. I think it just goes to show that there's always so much more that we need to know about the microbiome. So my title's called Microbiome, a piece of the personalized nutrition puzzle. And really it's called that because personalized nutrition is a very new field. It's very exciting, and there's many aspects that we need to consider to really be able to do this well and do a good job, because ultimately that's really what we want to do, is to be able to make an impact on people's health. So without further ado, really we're trying to unravel the variability in who we are, what we do, and why I'm making these choices. So this is actually seems very simple on paper, but actually is quite challenging. So what we need to consider is not only kind of what are biology is, so who we are as a person, and all those different parameters, which include the gut microbiome, but also what do we eat, and are we kind of looking at that in resolution and precision and depth to be able to really understand what the specific kind of individual reactions are. And then another thing that we really need to look at is also, it's not just what we eat, but also, how are we eating? Why making those choices? And what are the other different variables that are making a change or an impact onto our health and kind of different interactions that we might perhaps be looking at. So really, with this shift towards kind of personalized nutrition, we're shifting away from this kind of population-based guidance. But something I really want to highlight in this talk is actually we're not throwing population guidance out the window at all. It's very much that we are looking at precision nutrition and looking at whether there's certain aspects that we can kind of overlay over population advice. And I'll circle back to this at the end as well. So what we have the opportunity now these days is actually we can, we're actually at this time where we really can explore personalized nutrition. And really with the advent of big data and novel technologies, it's really allowed us to kind of collect that quantity of data, but also that resolution and precision of how we're looking at it, like you would with a traditional RCT, but kind of on steroids at the size that you would have traditionally in epidemiological study. So that really gives us this kind of big data and also these kind of machine learning approaches where we're really able to look at advice and look at it different ways. So, even if we're moving from general advice to then stratified advice, so stratified, we mean like we give advice to different population groups. So that might be cardiovascular disease, or perhaps, as Gwen was saying, kind of based on your enterotype, and then personalized advice is really kind of at that very top, which is giving advice specific to that individual person. So I want to talk a bit about the PREDICT studies, and really this is an ongoing program that is collaboration between tech and academia. So this company called Zoe that I work for is really founded within this collaboration that started with King's College London. And they spent, 3 years in stealth mode running research studies to be able to really investigate and understand what is happening in terms of people's responses to food. And then we collaborate with kind of various universities as well to really bring, access to those kind of remote trials, citizen scientists, and then collaborating with academia to be able to understand more about what's going on in our bodies. So we have multiple studies that we've run within the PREDICT series, and we also have a number that are ongoing. So I really want to highlight here, this is Dr. Sarah Berry's work, who's the lead investigator on this, and kind of is an expert in blood sugar and blood fat responses. And what we really started with was the PREDICT one, which was 1,100 participants who had, very in-depth dietary data collection. They came into the clinic, they had multiple tests, they took stool samples, so really collecting that really broad breadth of parameters. This happened to be within the Twins UK cohort, which is based at King's College London. So we actually had a number of twins there as well, so that we could look at, if there's any kind of genetic heritability factors. We've had multiple different studies going on, and at the moment we actually are on PREDICT 3, which currently we've actually recruited about 40,000 people who've gone through this program to date, and that just continues as we go. And likewise, we have some efficacy studies, because really we know that RCTs are kind of still the gold standard. So we're really kind of looking at that to see if we can get some more strength in terms of the causal associations there. So really one of the first papers that we really found was that there was significant variability between healthy individuals. And what I want to show is, again, that full picture of how different people are. And what we found was that there was kind of between people about 68% variability in triglycerides, and 40% in terms of glucose. But what we also saw was that within a person, if you look to the left at the very bottom here, we saw there was actually 36% of differences in their triglyceride response, and in terms of their blood sugar, there was 24%. So less than there was between two different people, but still some day-to-day variation. And this was in a response to kind of a tightly controlled situation where they had these standardized muffins, which were kind of metabolic challenge muffins. And we measured, their blood sugar and their blood fat response over a number of hours. What we then looked at was that we found that determinants differed for different outcomes. So if you look for glucose, actually meal composition at the very left in the purple was actually, one of the strongest kind of determinants. Genetics and meal context also played a larger role. Whereas for triglycerides, actually, it was the serum lipid markers and the fasting triglyceride levels and the habitual diet, which played a role as well. What you can see for both of them in the kind of blue color is actually the microbiome is at play, but it's kind of slightly smaller kind of proportions than some of the other components. And again, I just want to highlight, this meal context of that we still kind of need to know more about, that kind of gets often forgotten, which is this kind of time of day, the impact of exercise, meal ordering, kind of sleep, and also eating speed and more. So there's kind of a lot more to show there, and I think also shows how exciting this area of research is. So in terms of some of the microbiome research we've done with the PREDICT studies, we found that actually, looking at this PREDICT 1, where we had these twins, we found that actually only kind of 34% shared, the same species. And actually, if you're unrelated, you share 30%. So actually, despite this, we can see that even with the twins themselves, that there's kind of very different microbiome profiles. And what we know that microbiome is linked to kind of diet and health, and quite often we see that there's this kind of complex interplay that exists between the three. And we know that diet is, a very strong, modulator of the gut microbiome, with kind of whole foods that are rich in kind of microbiota accessible carbohydrates, and also polyphenols. The polyphenols are these bioactive compounds that are present in the color of fruits and vegetables. So actually, you get different types of polyphenols depending on different colors. And what they do is they tend to reach the large intestine, where the microbiota will process them and produce these beneficial metabolites, such as short-chain fatty acids. And it's these metabolites which then cross the body and have an impact on your health. So we wanted to kind of dig a bit deeper. So what we did was looked at microbes that are associated with kind of favorable or unfavorable measures of health and diet. So these are all kind of correlations between looking at specific kind of inflammatory and also kind of fat and blood pressure and kind of glycemic response measures. And what we found was that we were able to kind of identify these kind of selection of what we called kind of good bacteria, good species that were linked to kind of beneficial or kind of better levels of blood fat and blood sugar control. These people had lower inflammation, they had lower levels of body fat, and tended to have a better kind of cardiovascular and type 2 diabetes risk profile. Whereas we found with another collection of bacteria kind of the opposite. And what we saw was that these kind of gut microbes tended to influence glycemic and lipemic responses. And actually, in fact, that these were linked to diet. And we kind of see these kind of healthy dietary patterns and healthy plant foods really kind of associated with these kind of beneficial bacteria. So we looked quite closely to see a bit more at this relationship, and actually, yes, there's correlation, and I think this is something that it's really important to kind of communicate when we're talking about the gut microbiome, because it's only really in the last 20 years that we've made progress here. But what we can see is, for example, with yogurt, this is very kind of, we'd see this kind of example of Bifidobacterium animalis, so it's kind of confirms that there's kind of that signal coming up there. Likewise, we looked at, if we look at the top and the different colors, mainly in this kind of dark green color are the kind of foods that we would kind of assume as being kind of healthy plant-based foods, and also in the kind of lighter kind of yellow brown healthy animal based foods. So you can see actually there's quite a kind of neat difference between the top right and the red being kind of more strongly kind of linked to those foods that are typically kind of healthy animal and plant-based foods. As you can see here, for example, here we've got on the left, beef, poultry, sausages, pork and ham, so typically kind of less healthy kind of animal-based foods, whereas on the right we've got in the middle kind of shellfish, oily fish and eggs, which are kind of showing up kind of quite neatly within those groups as well. Something that we always like to highlight is actually red wine gets kind of deposited in the slightly beneficial group, and I think there has been some research about red wine being kind of linked to kind of gut microbial diversity and lower visceral fat mass, and that's kind of the hypothesis behind that is perhaps that it's very high in polyphenols because it's its dark red color, but I think still a lot more there to be understood. So again, just to kind of highlight that point about kind of correlation versus causation. So, it's very much, foods linked to higher levels of, certain bacteria. But I think the kind of way that we would want as kind of a field to be moving towards is really being able to look at this in a much tightly controlled, kind of randomized control trial setting, where we can see kind of very specifically that interventions of certain foods and food groups, we can see kind of specific changes that is consistent in different people, or perhaps within different stratified groups in terms of their gut microbiome. So I think this is an area that's only going to expand, and we'll find out more as we go. So why is it so hard to get a clear picture? I think, really it's this multiple factors. And I think, we all love a challenge. So I think, it's that gut microbiome is highly complex, but also diet is highly complex. And I really think that, diet has almost kind of been not really given a kind of full due in terms of, the potential analysis and techniques and the way of modeling dietary data that we apply kind of elsewhere, for example, with the gut microbiome. And then, of course, human health is also highly complex. And again, to kind of nod back to kind of Gwen's talk, there are other factors at play. For example, gut transit time that we should really be considering when we're doing this research. So interactions between diet, microbiota, and health. What we see is that, for example, we look to the left, that we have this individual eating pattern. And there's this direct effect on the host, which kind of decomposes into different chemical compounds and are absorbed across the gastrointestinal barrier. What happens also with these chemical compounds is that they are metabolized by the gut microbiota into metabolites, which then also can be absorbed across the gastrointestinal barrier and have an impact on host health status. So really, there's multiple things at play with different microbiotas, possibly, different species perhaps producing the same microbiota, same metabolites. So it might not actually even be that we should be looking just at the microbiota, but perhaps also considering the metabolome, which is this selection of metabolites as well. So we also have this microbial competitive and cooperative relationships. So actually within this actual community, there's kind of competitiveness and relationships between the same species, between different species. It's kind of like a kind of school where, there's different cliques that like to stick together, and perhaps, the jocks who are positioning themselves against, probably the science nerds on the other side of the corner. So then you have this entire community, but it's also the spatial organization. So how are the microbes kind of positioned together, some clustering more closely together, perhaps around a certain food source, and others also elsewhere. So that also is something to bear in mind when we're collecting these stool samples. You know, we collect, for example, a pea-sized scoop for some of them from, rather than a kind of a whole stool collection. And actually, are we just collecting a specific, you know, group that happens to be, you know, at that specific position, rather than representative of the entire stool itself. Just to kind of tie back also to this kind of characterization of dietary data. And I think this is something where we slightly fall short, and something where in the PREDICT studies, we really tried to, you know, really look at dietary data and kind of high precision and granularity, and collecting kind of nutrient information at brand level, for example. And really, you know, we've got these kind of nutrient databases, which, for example, for one banana will have one kind of nutrient profile, but yet we know that, for example, from being incredibly ripe to underripe, that there's actually quite a difference in terms of what happens. But again, also picturing food and the way that we eat it as humans, which is in kind of multiple combinations, you know, across the day, across, you know, a month, but also kind of that very granular shift, because we know the microbiome, you know, can fluctuate from kind of an hour to 24 hours, and we can see changes there. So does personalized nutrition have to be at the exclusion of population-based guidelines? Something I kind of want to share here is that, you know, of course, in some cases, you know, the average response is, you know, very meaningful. So, you know, in studies where we see that there's kind of an overwhelming kind of favoring over the intervention, you know, that really is a place where we really need that population guidance and population advice. But there are cases where actually the average response is perhaps a bit meaningless. And this is where, you know, in response to an intervention, you could have, you know, a group of people who favor the control and others that favor the intervention. This is where, you know, for precision nutrition can actually lay really nicely on top of the population-based model. Likewise, with the microbiome, we have this kind of generalized advice around, you know, more fiber-rich foods, you know, more polyphenols, you know, plant diversity, and actually, you know, is this place for kind of personalized advice where there's actually specific advice on top of that, where we can overlay that. That's really just advancing what we already know to the next level. So just to kind of tie that up, we're really in this kind of current phase of having this explosion of digital devices and remote clinical testing, and also, you know, able to, you know, use multiomics and citizen science to really understand perhaps what the multiple factors that impact our responses. And hopefully we can then shift to really, you know, giving specific kind of actionable advice to people about, you know, what, how, who, why, and feeding that into something that we can then, you know, be able to deliver in terms of impacting their health. And likewise, I really hope this has perhaps highlighted that, you know, the microbiome is really, you know, only one piece of the puzzle, but I think, you know, there's a lot that we will find out more with the microbiome. And I think there's a lot there that we can see that it does influence. And I think it's a very exciting area, as I'm sure we all think, because we're all here at this talk. And lastly, the very last, this is kind of a key message that my kind of science group has all decided that we want to share in presentations, is that we must not forget the pleasure in how we live our lives. And actually, you know, really it's about the kind of cultural, emotional, and social context, and enjoying foods together, and not to kind of, yeah, I think put everything into perspective. Yes. Thank you very much. And I want to say thank you to everybody who we work and collaborate with. And yes, if there are any questions, happy to answer them.
Thank you very much, Emily, for this presentation and the overview. I don't see any questions specifically in the chat yet, so I will just maybe, while giving participants the time to maybe add their thoughts there still. I'll just let me ask one quick question, maybe in terms of data sets to complement the gut microbiome data for the interpretation on health. So you have mentioned, of course, dietary intake. We have heard on transit time, for example. Can you maybe just briefly comment on what do you think would be most important other parameters, like, you know, genetic predisposition, dietary habits, environmental factors that should be collected to complement these insights from gut microbiota data for the interpretation, and how that should be integrated for really that more holistic view?
Yeah, I think what we really need to do is really understand what are the key other factors and contributors that are kind of influencing what's happening. I think, you know, Gwen's talk earlier about, you know, gut transit time is something that, you know, is, you know, he's got some great papers out there. We've done some research as well in terms of using kind of blue poo and kind of measuring how long that takes to kind of go through your GI system. And I think it's really making sure that we have kind of standardized approach across the research field to include these different factors. So I think, you know, we know that, for example, you know, gut transit time, great example, but also things like, you know, food processing level, should we be considering that within, you know, the dietary data that we're collecting? You know, timing of meals, really important. You know, that actually the circadian rhythm, we're seeing a lot more evidence coming around, for example, about time restricted eating. And in fact, you know, possibly, you know, this kind of conundrum of kind of skipping breakfast or not skipping breakfast on your health, you know, is that actually more related to kind of having your feeding window and your gut microbes, you know, to be determined. But I think really what we need to do is really understand what are the different factors at play, what are the main contributors, and really then adjusting and making sure we have a standardized approach in terms of the research community of including those key factors. And therefore, you know, having this, if we have a standardized approach, hopefully we get some more kind of standard homogeneous outcomes rather than this kind of a lot of kind of variability between research outcomes.
Yes, thank you so much. There is another question from the chat now. Maybe we can see how far we can get just as a very brief maybe answer, and otherwise we can take it also into the discussion later on, because there are more questions coming in now. Let me just add one more maybe for now, and then we will continue with the third presentation. The question is, population-based recommendations are based on leaving no one behind and should be seen as preventative recommendations. Thus, RNA or RDA levels are meant to avoid deficiency at the population levels, not the individual. It's a bit misleading that RNA, RDA values are taken as individual values when they were not designed to do this. Interesting to talk about personalized nutrition, but what is missing in the discussion is how will this work to replace the current guidelines? More importantly, how will this improve public health more than the current guidelines? I'm not sure if this question is too big to answer now. Otherwise, we can also circle back to it after, towards the end of the webinar. But maybe Emily, you just want to say a couple of sentences to answer.
Yes, I mean, I think this is exactly, you know, I think we all need to put our heads together and figure out how we can move forwards with this together. It is a very new field. You know, I think we really need to put our hands up and say we don't know everything. We don't know the answers to everything yet. And I think we do need to consider how this might work to be accessible for everybody, because realistically, this is something that isn't for the general population, specifically lower, you know, socioeconomic groups, which we know that actually making dietary changes would be the most impactful for their health, and kind of how can we support that in terms of public health measures. You know, I think we do see, you know, the population guidance, I think, gets a lot of flack, and actually, as you say, you know, it's based on, you know, leaving no one behind and, you know, preventative recommendations. So I think moving towards stratified recommendations, so like I mentioned at the beginning, where you have different kind of population groups and you could have kind of a specific guidance for those population groups, I think is the way forward to make it then more accessible for different people. And I really see that actually kind of the end of it, where we do all these different kind of parametric tests, you know, in an ideal world, you know, that would be incredibly cheap and everybody be able to access it, have it be to be accessible for everybody. We're just not at that stage yet. So I think it's really, you know, what are the steps that we can take towards that, and to bring everybody along on the journey with us as well.
Yes, absolutely. Thank you. I'm very sure that we will get back to that part of these aspects also in the discussion later on. Thank you so much, Emily.
Thank you.
We will continue with the third and last speaker for today, Dr. Sean Gibbons. Dr. Gibbons is an assistant professor at the Institute for Systems Biology and affiliate faculty in the bioengineering department at the University of Washington. He's the ISB Washington Research Foundation Distinguished Investigator, and his lab develops experimental and computational approaches for exploring and manipulating host microbe ecosystems. It has been a pleasure to work with Sean and the expert group for this ILSI Europe activity, of which Sean will present the output today in the presentation. And this is titled Leveraging the Gut Microbiota to Predict Personalized Responses to Dietary, Prebiotic and Probiotic Interventions. Sean.
Thanks so much, Gabriela. And huge thanks to Gwen and Emily for those excellent talks, kind of giving us background on the environmental factors that are shaping this microbiome that we're hearing so much about these days, the clinical associations that exist, which there are hundreds, and then Emily giving us a really interesting real-world example of how currently in their academic partners. So yeah, to build off of what Gabriela just said, I was kind of brought in to ILSI to work with a group of academics and industry scientists. This is just an overview of the members of the group. Source from the probiotics and prebiotics task forces, which we try to be more than 50% non-industry scientists. And so there are, we will also kind of telling you that the experts were not paid for the work, although academic members were given small honoraria. And if you want more information, you can reach out to ILSI about anything you see here today. But yeah, we sort of looked around at what was going on in nutrition and the microbiome and healthcare, and tried to see, what were the tools that were available for leveraging what we understand to be a high degree of heterogeneity across the human population and how we respond to many different interventions. I mean, it's true that population scale interventions are often very good for most people, and some are less so, but there's always people at the tails of the distribution who are not taken care of by these population scale interventions. And so I guess a lot of what this field is about is trying to figure out how do we help those folks. So, I come at this as a microbial ecologist. That's my background. I used to study like rivers and oceans and things like that. And now I'm a poop guy. And I think, the microbiome, as you learned from Gwen, has really kind of reshaped how we think of ourselves as an entity. We're not just solely human. There's about 100 times, more than 100 times the number of genes in our metagenome or in our microbiome than there are in our genomes, right? There's millions and millions of bacterial genes in our bodies and only 20, 30,000 human genes in our genome. And we've outsourced a fair amount of the functionality of our bodies to this internal ecosystem that resides in our colon. And the genome is a filter on how we experience our environmental exposures during the course of our lives, whether we get sick or whether we stay healthy when we're eating a certain diet or exercising at a certain level or what have you, right? The genome is a filter on how we experience these exposures. And we're coming to understand that the microbiome is another one of these filters. It affects how we absorb nutrition. It affects how we metabolize drugs. It influences all the systems of the body, specifically through circulating metabolites. Our group is just about to publish a paper actually where we try to partition the variation in the blood metabolome that's being explained by the genome and the microbiome/diet. And to our surprise, and I think a few groups are coming out with similar papers recently, if you looked at about a random sampling of 1,000 blood metabolites, you find that 60% of those are significantly associated with cross-sectional variability in either the genome or the microbiome. Then if you zoom in on those, 70% of those associations are solely driven by the microbiome. And then another 20% or so are hybrid associations where the genome and the microbes seem to be controlling to some level the levels of these analytes in the blood. So, that's just sort of astounding, the amount of control that the microbiome has over circulating analytes in the bloodstream. Say that the diet is almost part and parcel to the microbiome because they're so coupled and they're so correlated with one another that when you're quantifying the microbiome or when you're quantifying diet, a lot of the variability that you're quantifying on both of those in both of those systems is essentially overlapping, not completely, but to a large extent. So we wanted to think about how, sort of the future is now, I think, in a sense. You saw that with nutrition is a very young field. Precision medicine is a very young field. And there's been astounding progress in the last few decades. It's not in the clinics yet, right? It's not widely available yet, but I think the papers we see coming out have been pretty surprising and pretty interesting. And so we decided to look around at methods that were either in silico, essentially computational modeling, in vitro, tools that we could use in vitro to assess inter-individual heterogeneity, or in vivo, population trials or animal models, and how these tools are all being leveraged to study this phenomenon where you feed 20 people a banana and you get different chemicals coming out the other end. And that's just the truth. If you measure it, you see differences. And so how do we predict those differences? I'll just give you a quick example here on the right-hand side. This is a trial, a feeding study that was conducted by the Sonnenberg lab at Stanford, where they fed folks either a high fiber diet or a high fermented food diet over the course of several weeks. And in the case of the high fiber diet, they saw this really interesting heterogeneity. Generally speaking, you think that feeding people a high fiber diet, who are Americans, essentially, their background diet is kind of this developed world, high animal-based diet. You feed them all a high fiber diet, you expect them to get healthier. But over a range of markers of the immune system, inflammation markers, they find that there's a subset of these folks who actually have increasing levels of inflammation, bloating, and discomfort while on a high fiber diet, while 2/3 actually get better. They see reduction in inflammation. And so, how do we explain this type of inter-individual heterogeneity to the same exact intervention? And so, just rushing through this because we don't have a lot of time. For in silico, we sort of parsed it apart into a couple of different categories, right? You have the typical statistical modeling approach, or machine learning, if you want to call it that, or AI, if you want to call it that, where you're taking sort of deeply phenotyped populations where you have coincident data from both the microbiome and from, say, host physiology or host genomes or whatever. And you're assessing how folks are responding to an intervention. And this is a very famous example, as you'll see on the right. This is ZB paper from a few years ago, and which gave rise to a company similar to Zoe, right? This is a company called Day 2 has now emerged from this study, where the authors found that in this Israeli cohort, you could assess people's responses to dietary variation, looking at post-meal glucose spiking, and they found that with a number of clinical parameters, including data on the microbiome, you could fairly accurately predict someone's response to a meal, their glucose response. And you could even design meals, which you see down below here, that were either designed to be bad meals that were supposed to induce a kind of pre-diabetic, bad glucose response, and a good meal that's supposed to, reduce that level of glucose spiking. And the predictions worked well, and they matched sort of what you get if you did expert-based sort of interventions. And so this was sort of one of the big proofs of concept, I'd say, in our field. The advantages to this kind of a method is if you don't know how things work, if you don't have any mechanistic insight into what's driving what, you can collect a bunch of data and train a model on it and make predictions. And those predictions work pretty well as long as you're predicting on a cohort that is fairly similar in their molecular phenotype to the cohort you train the data on. The disadvantages is that these models are often kind of mechanistically opaque. A random forest, if you kind of looked inside of it, it's kind of hard to know what's driving what and then how it all works and how the prediction is actually made. And the predictions are only as good as how inclusive your training cohort is. So if you're predicting on individuals that look different from the folks you trained on, you're not going to get good predictions. The next example of a kind of modeling approach would be a mechanistic modeling approach. And this is actually something that my own lab works on. We're sort of at the interface of systems biology and the microbiome. And so this is just one example of a model we've built called MyCom, which is a flux balance analysis model we've extended FBA from single strain models to diverse multi-species communities by doing a few tricks. This is developed by Dr. Christian Wiener in my group, along with student Nick Bowman. And it allows us to sort of simulate the metabolic outputs of an individual's gut if you know the initial conditions of what the composition of their microbiome is, and if you know the composition of their diet, or if you have an approximate idea of their diet. And just to give you an example of how we've tried to test our modeling platform and see if it behaves itself and makes reasonable predictions, this is that dietary intervention study from the Sonnenberg lab again. And we wanted to ask, could this mechanistic modeling platform maybe give us insight into why these people are different? Why are there these different groups? And indeed, if we predict the amount of butyrate and propionate produced across these groups, we find that the folks that showed the higher levels of inflammation are predicted to produce less butyrate than everyone else in the population. And so we suspect that short chain fatty acid production may be an axis of explanatory power in this particular cohort. The advantages to these models is they don't need training data. They're mechanistic. You can set initial conditions, but you don't need to sort of train the model. They're transparent. They're an open book. You can look inside the model and see that this metabolite is being eaten by this bug, which is using it to cross-feed this other bug, this other metabolite, and you can trace the paths of all of that. The downside is it requires really detailed mechanistic knowledge about the system. So you need to know all the enzymes and what metabolites they take in and put out. And if you can't annotate certain genes or certain enzymes in an organism's genome, well, you're going to be missing those metabolic reactions in your modeling. So it's really hard sometimes to incorporate uncharacterized or unknown phenomena. And some people are working on hybrid approaches that combine statistical and mechanistic, but that's kind of hard. On the in vitro side, there's a lot of ways to go, but I'll just give you a few brief examples. You can actually create a little model of a person in vitro with these ex vivo batch culturing systems. So you can take poop from a person, you can make it into soup, and you can put that into all kinds of different flasks and do different interventions like adding inulin or tryptophan or whatever metabolite you want to add to the incubation, and then time series metabolomics to measure the fluxes of what metabolites are consumed, what metabolites are produced for an individual person's microbiota. Similar experiments can be done in continuous culture systems like this SHIME system here that a lot of people use. These are fairly cost effective. They're fairly high throughput, although SHIME have application, and you can do kind of arbitrary amounts of spatial and temporal sampling in these systems. The downsides to these in vitro models is, of course, with the greater control, you have lack of maybe reality to some extent. Certain bugs can't be cultured in these systems, and you're lacking often interactions with host tissue because the host is not involved in these models. You can kind of get a host side to these in vitro systems by adding these sort of layers of epithelial cells, like these guts on a chip systems that lots of folks are developing. There's a high level of control again in these types of systems, and you do see a representation of host metabolism or host tissue, but it's pretty high cost still. It's hard to make these things. It's hard for them to be made in any arbitrary person's lab. They really are made by specialists, so you sort of have to collaborate with a particular person who's good at making these things. And the flow rates that you see in these types of systems are just way higher than you'd actually see in a real world gut. So there are of course caveats to these systems as well, but it does allow you to personalize an in vitro model where you can put the stool from an individual person into these systems and see how they respond. You could even use epithelial cells, like take a biopsy plug and use those cells to populate one of these systems. Beyond in vitro and in silico, there's of course in vivo. Non-human animal models is sort of the first category I'd put forth. You have a lot of experimental control, particularly even over genetics, say, and you can have germ-free animals, so you can get at causality, and they're very useful, except oftentimes the physiology or the anatomy doesn't quite match the human system. So mice aren't necessarily the best model for the human anatomy, but I say pigs are closer, or primates. But then the ethics of using these organisms in research it's getting more and more difficult to justify using these kind of larger, more intelligent species. And I think industry is also under a lot of pressure from interest groups to reduce their usage of animal models. So then finally moving to sort of the peak of what can be done here, and this is what Emily was showing with what Predict is doing, is to use human populations, try to just directly measure things in the human system. It's the highest possible level of translational relevance. You have your classic randomized controlled trials, which are really designed for testing population scale interventions. You have more exotic intervention trials like crossover trials, like you see down here, where you can have multiple interventions that are switched between and kind of randomized in the order that they're given to people, and you can use these to allow to have a single person kind of act as their own control, essentially. And these are better for testing maybe not precision or personalized interventions, but at least maybe multiple interventions, and choose between a set of 3, 4, maybe 5 interventions that are optimal for different individuals. And so, yeah, the question is, can we leverage all of these types of tools to build engines that can make these precision predictions that truly have a beneficial effect for the individual? This is just a cartoon from a recent perspective we put together, but you could imagine having a population of folks that maybe they have high levels of systemic inflammation, and you'd like to just turn up butyrate production in that population. But it may not be the case that you can give them all the same exact intervention to achieve that process, right? Some people can't produce butyrate because, well, maybe they lack a lot of the metabolic capacity to make butyrate. Maybe Faecalibacterium prasenciae has gone extinct in their guts. Or maybe they just need to eat more dietary fiber. And so which intervention you give which person will probably vary quite a lot. To give a quick example of some of the stuff we're playing with our metabolic models in this precision space, one thing we can do is sort of model engraftment or species interactions in these models. So this plot here was generated by Alex Carr, a student in my lab, and it's a single person's microbiome. And these are all the different genera that are living in that person's microbiome. And what we've been able to do is just knock them out one at a time from the model and see how every other species in the system responds, right? If you knock one bug out and everybody else's growth rate goes up, well, that bug was suppressing the growth rate of other things in the system. Or vice versa, if you knock something out and some other bug's growth goes down, well, there was some sort of cooperative interaction going on. And we can see that for some taxa, like Bacteroides, overwhelmingly you see competitive associations with many other taxa. So if you knock it out, other things can grow faster. But other bugs that you might expect, like Akkermansia mucinophila, if you knock it out of the system, well, you get less mucus degradation and all the byproducts from that. And so a lot of other taxa actually grow more slowly in the system if you knock it out. And you can do a knock-in experiment as well. So you can actually take a bug that wasn't already living in that person's microbiome context, and you can put it in silico and see if it can grow. And right now we're working with C. diff as a proof of concept to see if we can predict colonization or engraftment in an individual's microbiome. And this is what Alex is working on. Just some really quick prelim data from that. You can look at a population with varying Shannon diversity. So we often think high diversity is more protective against invasion. And indeed, we do see that the average predicted growth rate of C. diff when you invade these different models with C. diff is highest in the lowest diversity folks. But we also actually kind of see a bit of an uptick when you get towards really high diversity. And probably individuals who are constipated are often in this higher tail here, as Quen showed us. We can also take data where we have a known introduction event of C. diff. So this is a time series from Eric Alm's lab, this David et al. Paper, where the individual got food poisoning around where this red spike is. They got a salmonella infection, had a bad piece of French toast. And okay, yes, my 2-minute warning. Sorry, I'm just checking. Okay, sorry, I'll try to wrap this up. And so we know that C. diff was introduced during this food poisoning event, and this is what the blue trace is here at the bottom. It's the kind of quantified amount of C. diff in the system, very low, but it's detectable here. And we tried to invade this this time series with C. diff, and throughout the whole time series, we couldn't get it to grow, except at this one point where you see this introduction occurred. So we think this is going to be useful for predicting engraftment of probiotics or for FMT engraftment predictions, and in addition to predicting things like butyrate production and so on. So at the end of this paper that we put out, this perspective, we have a brief section of imagining a future healthcare clinic that's kind of outside of the norm of what we have today, which is, you have a symptom, you go in and you treat the symptom. You get a pill or whatever to get rid of whatever symptom is causing you trouble, but you might not be addressing the actual underlying cause of why you had the symptom to begin with. And so perhaps the future should involve more quantitative phenotyping, getting genomes and metabolomes and so on and so forth, tracking people over time and trying to provide them with a little more TLC and personalized care. Of course, this is also a quite much more expensive way to provide healthcare, but we're hoping that in the future we can make these things cheaper, especially through using more in silico approaches. So I think I'm kind of out of time, so I'll skip all of this. But I think Emily hit the nail on the head earlier, saying that it's complicated, right? These systems are complex, and Gwen also mentioned dysbiosis and how some people have broken microbiomes, and I would throw an even further wrench into that and say that some microbiomes can be good or bad depending on the context, right? There's some evidence showing that people who are high Prevotella, if you feed them a high fiber diet, they actually do better than other people as far as cardiometabolic markers and weight loss. But that those same high Prevotella folks, when fed a sort of Western diet or a developed world diet, they actually get less healthy compared to everyone else. They see worse lipid profiles and so on. And so it's a sort of effect multiplier. You do better in one sense and you do worse in another sense. And I think that's true for a lot of these things, that context is key and that we're just at the tip of understanding it all. So with that, I think I'm over time, so I'll end.
Thank you very much, Sean, for this presentation and the overview of the activity and the publication. I will just start with two maybe more technical questions to your presentation, and then we can open up for the discussion also with all of the three presenters, I hope. There's one question, thank you for the interesting talk. How good can the in silico models be if no quantitative data, such as real number of bacteria in the pool or sample, are available? 99.9% of all microbiome data sets just comprise relative abundance data. Would you like to comment on that?
It's a good question. So in our metabolic modeling platform, we actually provide initial conditions on population sizes, and often we're using relative estimates. But the model itself sort of readjusts the abundances of those populations in order to make them fit together well, as far as who's cross-feeding who and what's eating what. And at the end of that, we tend to get estimates of abundance that are more quantitatively related to when we do have measures of total biomass to those measures. So we think that our metabolic model actually helps us to better infer from relative abundance what the true estimates are. But yeah, that's definitely an issue, and we should do a better job of making those measurements, I think.
Yeah, thank you. And I think there's been one other technical question to the work from Alex Carr that you presented also on quorum sensing. Have you been looking at that molecule formation in the work from Alex Carr?
On quorum sensing. So, well, the model I presented, MyCom, is a FBA, flux balance analysis, and those models only contain information about enzymes and metabolites. And so quorum sensing, it doesn't fit into that rubric. It wouldn't be taken into account. So anything that has to do with like that kind of communication or toxicity, like oftentimes microbes try to poke holes in each other's membranes, those types of interactions are not captured by the metabolic modeling framework that we're using, and that anyone who does FBA uses. But there are ways of building in those types of interactions, and we haven't explored that.
Okay, thank you very much. Thank you to all three presenters for your talks and already answering some of the questions. I would like to maybe just for the last part of the webinar, just open up for the discussion with the three of you there. And probably one of the most important aspects to come back to is probably really to how can we move forwards in terms of some of the steps mentioned already towards application of these approaches. How can we really make sure that some of the methods, as you look at some of the questions in the chat too, how can we make sure that some of the tools that we are using maybe for research purposes right now, that this would really be more cost effective, and also in terms of practicality, in terms of using really from a consumer point of view and really having these applications in this future healthcare scenario that we are thinking about. I'm not sure if anyone from the three of you would like maybe to give that a start in terms of thinking about some of the visions you might have in terms of how do you see this becoming a reality also from a application and consumer perspective in the end.
I'm happy to jump in. I think we've had some experience with. I think even what we've done as a company at SPE has just showed how basically our test kits cost about £1,000, which was effectively the price, and when we were offering it at a much lower price to customers. And I think even when the time the company's been running in the last couple of years, that price has kind of decreased drastically in terms of how much it costs for us to do it. And I think that really just harks back to it's really that basics of kind of supply and demand. Inherently, if there's more people doing microbiome testing, more people pushing the kind of frontier forwards, we will be able to access kind of better techniques. For example, think about 16S versus shotgun and how now shotgun metagenomics are kind of used much more widely, whereas before it was incredibly expensive. And I think the same for if we look at measuring those metabolites, that actually that is quite expensive. It's not something that we can really deliver to users now, but actually who knows in a couple of years time that might actually be something that we will have access to, and hopefully it'll get to the point where it's something that you have free through your healthcare system. And I think it's really about having the foresight to look ahead and believe that these things are possible and that we need to work our way through towards them, rather than kind of think, oh well, this is just not going to work, or how much evidence is there around this. Inherently, we've got to keep building the evidence, and we see there's lots of great signals for that being a great possibility, but also doing it in a way that is kind of ethical. And I think one of the participants made great suggestions, saying that we need to be asking the right questions, which we do. We need to be making sure we're using this data to the best possible way and thinking about how it can be actionable for people. I think that's very much with predict research that we're doing at the moment, is this a way that we can give specific advice from the specific studies that we're doing? So a lot of that is also how we're phrasing the questions, as well as considering all these different parameters, and thinking about doing things in a very kind of rigorous methodological way as well.
Yeah, mind if I jump in, Glenn? I'll maybe take the next one. So the way that's great, Emily, and the way I thought about it at our institute is to understand the heterogeneity that we're seeing in these responses, you almost have to spend a lot of money up front, sadly, to really deeply quantify folks. You measure all these parameters about their bodies. We're trying to push for smaller trials maybe, but just collect a lot more information longitudinally about people, where even if the intervention or what have you, it fails, or it's very heterogeneous, or it's all over the map, at the very least you can go back and look at the groupings of people and how they respond, and partition them based on these molecular phenotypes to understand mechanistically what might be driving these variable responses. And once you have enough data from these larger population, smaller populations, you can start to pull out, maybe we only measure a few things to actually get a handle on those groupings. We can make these sort of measurements cheaper and higher throughput by doing that. And I think another thing is we should think carefully about making these things more democratically available and more cheap. at the onset, and try to incorporate things that are cheap to measure, like wearables or sensors that you can easily give people that they can move around with, that are low cost, that we can try to infer these other measures from. If we can project expensive measures into some sort of cheaper space, we can make these things more available.
Yeah, thank you for these comments. I'm not sure, Gwen, from your perspective, if you also want to comment on that specific question, and or maybe also, I just want to bring also up to you maybe one question that has also been added in terms of for some areas, where would we think that this would be mostly relevant? Maybe there's a question, would you recommend for people with health issues or discomfort, or people in general? You have been presenting on some work on, for example, inflammatory bowel disease in your data, I believe, I recall from your slides. Are there specific areas where you think that it would be mostly where the area is already more developed, or where there's where there might be more, where it might be most promising maybe to focus on also moving forward?
So with the work that we are doing, I mean, there are two roads that we're actually interested in. And one would be more preventive healthcare, to give advice to people on how to change lifestyle, diet, or whatever, in order to keep their health. The other would be like therapeutic interventions. I think that indeed what we are proposing, or the tools that we are using for the moment, that they are very costly and that they are not very democratic, or that not everybody has access to them. I agree with Sean that we should look for simpler tools, and I think there will be possibilities also that we don't need to do all analysis with all patients in future. We just need to determine those targets that are of interest. But also, I mean, preventive healthcare saves a lot of money. Yeah? The budgets that are used in health insurance in Europe, that we are spending on anti-inflammatories, on proton pump inhibitors, on before it was cholesterol lowering medications, statins, and so on. These are huge budgets. So it's just a shift towards preventing and not to well, it's from managing to preventing, actually, because we're not curing and we're managing disease in most cases. So I think there is a lot to be gained for a society, also talking in terms of budgets, actually. Now, the second question was on where do I think that the applications are, or where could we have applications in future? Did I understand correctly, Gabrielle?
Yes, maybe some of the areas where it might be most promising so far. From
I think we're most advanced, of course, in the field of IBD, because we did most of the studies there. And with IBD also there, like, for example, one of the things that we hope to get rid of, and I think we're advancing towards that soon, is the cascade of different therapies, and where we say, like, we're going to treat you with this therapy, and if it doesn't work, you come back and we're going to prescribe you another therapy. And so there, we think that and many people are moving in that direction, many groups are moving in that direction, that may be based on several factors, including the microbiome, because microbiome is definitely not the only player there. We hope to be able to target people towards the most efficient therapeutic route for them from the start. And I think that's, well, it will benefit definitely to the patients, and it also will save the healthcare a serious cost, because this trial and error based way of working is also, well, it doesn't help the health insurance and doesn't help the patients. So I think there we're closest to moving forward, actually.
Yeah, thank you for these thoughts. I think there's also maybe just one comment further also on how different data sets can be integrated, and the learnings from different studies and data sets also to be combined for actually all of the learnings, and really comparing data sets, but also building on each other in terms of the learnings there. I'm a little bit afraid we are running out of time, because we only have 1 minute left. I think there was a question from Ilse. Let me just check with the comment. I think we just have 1 minute left for wrapping up probably the discussion. I think probably in terms of how to best integrate the data and how to propose steps moving forward, actually, I'm not sure if any of you has one kind of final thought or one statement that you would want to share, maybe just before we are wrapping up the session, in terms of most important remarks on steps moving forward and integration of the data.
Maybe I'll quickly
Famous
say,
last
one thing,
words.
one thing we've noticed is like making people participants in their own healthcare increases their engagement, their compliance, having more control over your own health trajectory. Maybe not for everybody, but for a lot of people is a valuable thing. And I think healthcare isn't designed to be participatory. It's more like the doctor tells you X. And I think these precision approaches sort of open the window towards individuals having more control over their own health and their own healthcare, which I think is great.
Yes, I think these are beautiful, beautiful words to wrap up the session. I'm afraid we are running out of time. Thank you all for your questions and comments. If you have anything else to add as comments or feedback, please don't hesitate to reach out to ILSI Europe. We will be happy to kind of channel the questions back to the speakers to also come back to you. Thank you all for your participation today. A big thank you to the 3 fantastic speakers and all the preparation, and ILSI Europe also for the organizing the webinar. Thank you so much for the discussion. I hope the field is gonna further evolve with a lot of exciting developments also moving forward. Thank you very much again, and I hope to see you soon again. Have a very good day. Thank you so much.
Thank you very much, Gabrielle. Thanks.
Yeah, thank you.
Thank you all.
Thanks, Gabrielle.
Bye-bye.
Thank you.